Thank you all for coming this evening. My name is Teddy DiLorenzo. I'm the Interim Associate Dean for the College of Behavioral and Social Sciences. I'll be making a few introductions, but before I get started, I'd like to invite Dr. Eddie Vela, the Dean of College of Behavioral and Social Sciences, to come up and extend his invitation, his welcome to you and make a couple of introductory remarks. Thanks.
I sincerely want to thank you guys for taking time to come out tonight. I know it's chilly, but this is a fabulous evening and one of my favorite, actually my favorite event in the fall semester. There are, of course, some people I want to thank just very quickly. There's no way this could happen without the work and the dedication of lots of people, but especially our Associate Dean, Dr. Teddy DiLorenzo. So please give her a hand. She's our new dean. She's been at this just for a few months, and she steps into the door, and I say, Teddy, I have a task for you. And so she did it, and she did it well. And of course, she had a lot of help from Kathy Castleberry, who's done yeoman's work in getting this off the ground. So there's Kathy there, so thank you. This is, I believe, around the 10th year that we've had this event, and I've been to the event every year. 10 years ago, I was a faculty person and not doing administrative stuff. And I'll tell you something that I don't think anybody knows. The very first year that I was here, that I attended the event, there was an interim dean, whose name shall remain nameless, who was in the spot. And I was talking to this person after the first colloquium, and he told me, did I say he? The interim dean told me there was an administrator that he was having a conversation with. The administrator told him that, you know, the College of Behavioral and Social Sciences, well, you guys are a service college. You don't have very much research going on. And other colleges really are doing the scholarship. And so what the interim dean decided to do was basically to start this event and say, really? We have some fabulous researchers here, and we've got people doing some marvelous work. And so what this event showcases is just a few of the research projects that are going on campus. The poster sessions, the posters that you see are just a very few of the marvelous research projects that characterizes and epitomizes what's happening in this college. Most of it actually in collaboration with students, which is I think that's what characterizes this college. And of course, we're going to be able to hear tonight from one of our superstars in the college, Dr. Bartelink, and I personally look forward to it. Speaking of Eric, there are a couple of reasons why I like Eric, but just a couple. One, he's really big. And a really nice guy. The other thing, he came to us in 2006 from my alma mater, Texas A&M, and so we're both Aggies, and so we had something in common. Both of us agreed that we were Aggies by geography, not by philosophy, and we just happened to go to school there. And but we had a lot of things in common. Eric has distinguished himself through the years. He got early tenure. In 2009 or thereabouts. Am I stealing your thunder? Yeah, okay. Sorry. I'll let Teddy tell you all the awards and accolades he's received. His students love him. He brings his students, he involves his students in all his work, both in his research and his professional activity as a forensic anthropologist. And so anyway, I want to thank you, Eric, for everything you've done. I'm really glad. I think your selection tonight as our keynote speaker is right on the mark. And I'm, as interim dean, I also know that there are lots of fabulous faculty out there, so we have an embarrassment of riches with respect to people in the queue who are going to be coming up down the road. And so in any event, I'll stop rambling here and turn this over to Teddy to do the actual introductions for Dr. Bartelink. So thank you all for coming out tonight.
Well, thank you, Dean Vela, very much. And thank you all for coming here. Again, as Eddie said, I know it's a cold night out, so it's very nice to see a lot of you here who could probably be elsewhere, so thank you for sharing your time with us. I will be introducing our speaker, but before I do that, I would also like to give some of my thanks. First off to Dean Vela. He was the associate dean before me for some time, and he was the one who put these events on every year. And as the acting dean now, he has continued to support it, and I think that's an important part of how we are so successful. So thank you, Dean Vela. Also thank you to the faculty who presented today. All of the posters in the back do represent some of the best scholarship that we are engaging in the College of Behavioral and Social Sciences, and it took some time and effort to put the posters together. So thank you, faculty, for putting those all together. I also want to thank Kathy Castleberry, who's there, who deserves another round of applause, please. It all looks great in great part because of Kathy. She's behind the scenes and doing everything, including all the dotting the I's and crossing the T's, and I really couldn't have pulled this off without her, so thank you very much. Also want to recognize and thank the student ambassadors. We have some of our student ambassadors here. Some of them helped us haul the big poster boards over here from Holt Hall. So all the way along the line, we've had some great student ambassadors helping us out. I also like to thank some of the staff over in the basement of the library, Chris Ficken and Johnny Poon, and then we have, I think, Ben the photographer, who all have helped our faculty with their poster presentations, some up through, I think, this afternoon or yesterday afternoon. So we appreciate them being so flexible. They've been very supportive of the college, and so I appreciate that. As Eddie said, this year is the 10th year of the BSS Faculty Colloquium, and it is a forum where we recognize faculty, somebody every year who's chosen as a keynote speaker who has distinguished themselves in the wider academic community. Our past honorees have been recognized nationally as well as internationally for their scholarship and their work, and our current honoree certainly is no exception. We highlight proudly the remarkable accomplishments of Dr. Eric Bartelink. As Eddie said, 2006, PhD from Texas A&M, and he joined us as an assistant professor that same year. In 2009, and currently he's an associate professor. In 2009, he was recognized by Chico State's Faculty Recognition and Support Committee for his significant professional achievement. His current research is in human skeletal biology, and that research has led him into two related research fields, one in forensic anthropology, and the second, which is the subject of his speech tonight, is stable isotope analysis. Dr. Bartelink's research in forensic applications of stable isotope analysis can help determine the birthplace and migration history of unidentified individuals based on the chemical signatures in their bones and their teeth. He serves as the director of Chico State's Human Identification Lab, the hub of the forensic program. And the work that the lab does is they receive unidentified human remains from jurisdictions, Northern California and Western Nevada primarily for analysis and identification. During his time here, he's completed many forensic case reports, as well as supported increasingly numerous field research and recovery operation. He regularly supervises interns and graduate students and has helped coordinate the certificate in forensic identification. His extensive work with anthropology students has garnered further recognition for him by the Faculty Recognition and Support Committee, which honored him with the Campus Outstanding Research Mentor for 2011-2012. The recognition of his work has established him as an important contributor in such professional organizations as the State of California's National Association for Missing and Unidentified Persons and the American Society of Crime Laboratory Directors Laboratory Accreditation Board Assessors Workshop. He regularly collaborates with other universities within the CSU system, as well as the UC system, Cambridge University, and Southern Connecticut State University. He is an active researcher and has published in numerous journals. During his recent sabbatical alone, he co-authored a textbook, submitted 9 peer-reviewed articles that have been accepted for publication, completed 6 stable isotope technical reports, 11 forensic anthropology reports, and co-presented at 21 conferences, to name just a few of his recent accomplishments. After Dr. Bartelink's presentation, we will have some time for questions. So if you have some questions about his presentation, just let us know. We'll have one of our runners come find you. And with no further delay, I'd like to introduce and welcome Dr. Eric Bartelink.
Wasn't expecting all that. Wow. All right. I want to start by thanking everyone for coming. I know this is a terrible time of the semester to have to come out, especially on a cold night like tonight, and to listen to me talk when you probably have other things to do. So I really appreciate you coming out. And rumor has it someone's going to be pulling a lottery ticket out later for the best poster award or something like that. So stay tuned for that. Okay, so today I want to talk a little bit about sort of anthropological perspectives on how we reconstruct the diet of earlier humans. And so I'm going to have kind of a lofty goal of trying to cram 7 million years of prehistory into 50 minutes. So we'll see how successful I am with that. But I'm going to start with sort of talking about sort of why humans are designed to eat what we do, and then kind of shift to talking a little bit about my research in native California prehistory, looking at dietary reconstructions of Native Americans from the San Francisco Bay Area and also in the Central Valley of California. Okay, so we'll talk about this "Are you what you eat" throughout the course of this presentation. Okay, so why do anthropologists care about human diet? We know that anthropologists study human cultures. We know that anthropologists study prehistory. We know anthropologists study languages, and we know anthropologists study skeletons and DNA and things of that sort. Diet is a key factor in the evolution of the human species. So obviously it's going to be something that's going to be of interest to anthropologists of all different subdisciplines. And food is also a key aspect of culture. It sort of forms, who we are as people, and different cultures identify themselves based on what they eat. It's obviously very important, and what you put in your mouth is probably one of the biggest decisions you make every day. It also influences how we grow and age. So certainly the foods that we eat, starting from breastfeeding and infancy and shifting over to complementary foods and then eventually to a child diet and adult diet, those are going to affect how we grow, how well we grow. And we can see that in impoverished countries where children are growth stunted as adults. Okay, so it also influences how we age. If we eat a good diet and we're relatively healthy, we're probably going to age in a kinder fashion than if we eat a very bad diet. So it's what your doctor tells you, right, when you go and you tell him what you eat and he doesn't or she doesn't like what you eat. It also influences your health status. So certainly the foods that you eat can influence whether or not you're going to end up with certain kinds of degenerative diseases or cardiovascular disease, as well as other factors like genetics. Food also can tell us something about social status. And so culture anthropologists are obviously very interested in looking at social inequality and also what makes one individual more prestigious in a society than another. And a lot of that translates in many traditional societies, as well as societies of the past, into who gets the best food and who gets, maybe what's left over. Okay, so I did say I was going to talk about 7 million years of prehistory, but I'll do this very quickly. The human fossil record has really exploded over the last 20 years. We now have well over a dozen different species of hominins. So, humans, basically anything that walked on two legs that we identify as being part of our lineage. Some of them led to us, some of those earlier humans. Others are extinct sidelines on the human evolutionary tree. But we have basically 7 million years to work with when we're looking at how we became human and how our diet became what it is today. Okay, this is just a phylogeny, or sort of a hypothetical tree showing you relationships of different fossil hominins. We can start here at 7 million years ago. And if I can get this pointer working here. Guess not. Guess I don't have a pointer. All right. Basically, if we just if you start there on the very bottom, you'll see 7 million years ago we have a early fossil hominin called Sahelanthropus chadensis found in the Sahel Desert of Chad, Africa. That's the earliest thing that we think walked on two legs. It has a the position of where the spinal column is forward enough to indicate that the head probably bounced on a bipedal human, and we see reduced canines and some other features that we associate with our lineage. And you can track that all the way up to the very top there to Homo sapiens. And so you can see over the course of 7 million years, we have a lot of different morphological changes happening in the human lineage. And I'm just going to talk about some of those. How do we know what we think we know about the evolution of the human diet? That was part of the seminar I taught this semester, and I see there's several students from the class, paleonutrition. You'll see a photo of them later. This book is one of the texts that we use, and it's basically called Evolution of the Human Diet, and it sort of addresses the known, the unknown, and the unknowable, kind of looking at what can we say about early hominin evolution and what early hominins ate and what kinds of environments they adapted to. So basically this comes down to looking at their subsistence, and that can be reconstructed imperfectly from a number of different avenues. One is looking at basically the fossil animals and plants that they may have consumed or that lived in that environment, trying to reconstruct the environment that they lived in. Stone tool technology, so if they're using stone tools to process things like animal carcasses and things like that. We can also look at their skeletal and dental morphology. So there's going to be aspects of their bones and teeth that tell us something about, especially their jaws, what types of foods that they were eating. And then finally, we have stable isotope analysis, which is a technique that involves looking at the chemistry of bones and teeth, and that can be used to trace different kinds of foods that were consumed in the diet. Okay. If we go back to the very first discovery of an earlier hominin, we have to look at Raymond Dart here. He was an anatomist in South Africa who discovered the first Australopithecine. And this was discovered in 1924. And Raymond Dart was very controversial because he was trying to argue that this is evidence of early hominins in Africa. And it took several decades before this was really well accepted, in part because of the Piltdown Man hoax in England and things like that. But he really saw these hominids living in these caves, these limestone caves in South Africa, and that they were using the bones in that cave as tools. And there you have that sort of killer ape from 2001, and the sort of idea that, early hominins were very sort of aggressive kind of killer apes. We know that these bones that accumulate in these caves accumulated due to natural processes like animals, scavengers bringing carcasses in or things falling down into the cracks and crevices of these caves, and that these were not really tools at all. And he called this the osteodonto-keratic tool industry, which translates to bone, tooth, and horn. Okay, he had a very vivid imagination. He was a very good anatomist, but most of these tools really don't look like they were tools at all. They were just accumulations of animal bones. Okay, so we don't really have good evidence of sort of hunting behavior in early hominins like Australopithecines. Okay, so if we just look at sort of 5 broad trends in human evolution, and these I think are the major ones. We can look at things like shifting from a quadrupedal, walking on 4 legs type of locomotion to walking on 2 legs. So bipedalism seems to be the first major shift we see in the human fossil record. Then we have increases in brain size. So our brains are about 3 times larger than they should be given a primate of our size. So that requires some explaining. There is an increase in body size and a shift towards more modern body proportions. So from having relatively shorter legs and longer arms to having shorter arms and longer legs. There's a reduction in the size of the jaws and teeth, and also associated with a thickening of tooth enamel for eating more coarser types of foods. And then we have material culture. We have evidence of the first stone tools, the first objects ever made by our early human, dating back to about 2.5 million years ago. Okay, obviously these are all important parts of mosaic evolution of the hominin lineage. Okay, so we have lots of skeletal evidence of bipedalism by looking at the joint anatomy of early skeletons, and so that seems to be fairly clear. We also have fossilized footprints that are around 3 million years old from the site of Laetoli in Tanzania, fossilized in basically volcanic ash, and you can see good evidence of this bipedal locomotion going back 3 million years ago. Okay, look where our brain size is relative to other primates. Okay, down on the bottom part of the scale, you can see Pan is the chimpanzee, Pongo is the gorilla. Sorry, Pongo is the orangutan, and gorilla is the gorilla. And those are the genus names. And you can see Homo, our species, way out there like an outlier, statistical outlier. Our brains are huge relative to our body size compared to other primates. Okay, and we can track this through time. On the left side of the graph, you can see it's basically a time scale going back from about 3.5 million years all the way up towards the present. And you can see that those are just cranial capacities, which is a crude measure of brain size of different fossil human species. So as time goes on, we see these significant increases in brain size. Okay, so something is driving this change in brain size in the hominin lineage. Okay, that leads us to something called the expensive tissue hypothesis. And basically we know that meat is an important part of the diet of most forager societies. So people that hunt and gather for a living usually makes up around 30% of their diet. And modern forager societies consume typically nutritionally dense diets. And when you look at our different organ sizes, you can see that the brain is much larger than expected. If you look at on the right-hand side, and then on the left-hand side, you can see what you would, the left-hand side is what you observe, really large brains. On the right-hand side is what you'd expect. But you can see our digestive tracts are actually quite small, much smaller than expected. And so if you look at the two most costly organs in the human body, it's the brain and the digestive system, the GI tract basically. And it's metabolically costly to fuel both of those. So it's a trade-off. It looks like gut size, the size of our GI tract got smaller and our brains got larger. And there's quite a bit of support for this hypothesis. Our brain tissue is expensive. At 16 times more energy demand than muscle, requires 400 kilocalories per day just to spend on brain metabolism. And humans expend a much larger proportion of their resting metabolic rate than other primates and mammals. And that's just showing you brain size differences between humans and various groups of primates. And they're actually today more interested in counting neurons because that seems to be a better measure of brain complexity than simply just brain size. Okay, those are actually neuron measurements underneath there. So humans need much higher quality diets, so diets that are more dense in calories and nutrients. We also have a need to consume higher amounts of fat than other primates. We require some key fatty acids. So these are long-chain polyunsaturated fatty acids that are essential for brain growth in mammals. And I will probably butcher the pronunciation of these. Docohexacionic acid and arachidonic acid are key for brain development, especially in infancy and early childhood. And these are present in low amounts in plant foods, but seem to be abundant in things like meat and breast milk. Okay, so in order to feed that metabolically costly brain, we need to have adequate amounts of protein, especially during growth and development. Okay, so we see increases in body size and modern human body proportions. So on the left, you see an example of an Australopithecine, like those of you that are familiar with Lucy. In the middle, you can see Homo habilis, which is often touted as the earliest member of the genus Homo. And then you can see Homo erectus there on the right. And Homo erectus on the right is the first sort of human fossil that we see that starts to show body sizes and body proportions that are very similar to our own. Okay, and Homo erectus dates to 1.8 million years ago. That's the oldest evidence of Homo erectus. Okay, we know that our teeth are also designed to process different kinds of foods. So we have different kinds of dietary adaptations. Here you have frugivorous monkey eating fruit, that beautiful tarsier who's eating an insect in the middle, and omnivores on the lower right-hand side, chimpanzee. Okay, we know that humans in general are fairly omnivorous. Again, that's been a subject of a lot of discussion. But different animals have different kinds of teeth, different cusp patterns and things like that are designed to process a specific kind of diet. And some are more generalists. And so animals that are omnivores, so chimpanzees, humans, bears, raccoons, and pigs, all show some general similarities in their tooth molar structure for a generalized kind of diet. You're probably all glad you don't have carnassial teeth like a wolf or something like that. Okay, we see reductions in tooth size. Okay, so on the left-hand side there you can see human canines are very small. Early humans, like Ardipithecus ramidus, which is about 4.4 million years old, in the middle, and a larger canine on the right-hand side of a chimpanzee. So we've had a significant reduction in the size of the jaws and teeth, including canines. We've also had an increase in the thickness of tooth enamel, and that's a shift towards a more tough to chew types of diets that humans seem to have adapted to that make them different from apes that have very thin tooth enamel. Okay, we have evidence of meat eating and cooking. Okay, and like I mentioned before, the earliest evidence of any stone tools in the fossil record are about 2.5 million years old. They're found in East Africa. They're often referred to as the Oldowan stone tool industry after the site Olduvai Gorge. And so we have tools, the first evidence of tools going back to 2.5 million years ago. Some of these tools are associated with butchering animal bones, but a key factor at 2.5 million years ago doesn't seem to be hunting, seems to be scavenging. And so these early hominins from 2.5 million years ago, probably Homo habilis, were probably getting to animal carcasses after scavenging animals got to them, and were eating things like the leftover meat on the bones, as well as breaking the bones open to get the fatty, rich marrow that's on the inside. We don't see any real evidence of hunting at 2.5 million years ago, but recently there's been some studies in South Africa that have found evidence of cooking that go back about 1.8 million years ago. It's in, I forget the name of the cave. I think it's Waterswork Cave or something like that. They have evidence of cooking of bones and meat in that cave that go back 1.8 million years ago. And they also have some evidence of use of fire in Olduvai Gorge that date about 1.8 million years ago, and some evidence of using butchering animal bones that seem to be more consistent with hunting rather than scavenging. So while this stuff will be controversial, I think that there's some evidence to push cooking and hunting back into at least the time of Homo erectus. Okay, a lot of these early hominins, though, were probably more likely to be the hunted than the hunters. And here you can see a Australopithecine skull. It's actually of a child, And so an early hominin that was most likely eaten by a leopard, and you can see a reconstruction of how those tooth marks got into the bone. These were small-bodied hominins. They were probably, 3.5, 4 feet tall. Probably were not going to be very effective at hunting, larger animals. It doesn't mean that they weren't eating meat. It just probably means that hunting hadn't really developed at this point. Okay. We also can tell something about early hominin diets by looking at the chemistry. Now, these bones are fossilized. They're very old, and the organic material is pretty much gone. So you're not going to do DNA testing on an Australopithecine. But we can look at the tooth enamel, and the mineral structure often is still intact enough to get some kind of dietary information. So on the left-hand side, you can see two different types of plants, C3 plants and C4 plants. Those two types of plants follow different photosynthetic pathways. And so if you eat those different plants, those isotopes in those plants get incorporated into your tissues. So you are what you eat isotopically. Almost all plants are C3 plants, and so C4 plants are typically today things that are cultigens like maize, millet, amaranth, sorghum, sugarcane. Back in the late Miocene, early Pliocene, around 5 to 4 million years ago, we're having tropical grasses in Africa that are being exploited by some of these Australopithecines. And so you can see that some of those Australopithecines, their isotope values indicate that they're eating some C3 and some C4 plants, which was something that wasn't really expected. And this is very recent research because most people don't want to give you fossil teeth to destroy, to do chemical analysis on. We also have some information about Neanderthals, much later hominin that was around in Europe, the Middle East, and the Near East during the Middle Pleistocene. And they seem to be top level carnivores. They're clearly eating the nitrogen isotopes that are on the y-axis are elevated above different kinds of game. So you can see bison, cervid, herbivores, cave bear, etc. They seem to be enriched above that diet. So Neanderthals seem to be, consistently all the way across the span of Europe into the Near East and Middle East. They seem to be top-level carnivores in some regard. And we also see that they're distinctive. The ones in red are Neanderthals, and the ones in blue are modern humans that date to around the same time period. These are roughly 30 to 40,000 years old. And you can see there's some distinctions between modern humans and Neanderthals that were living often in the same general locations. Okay, so we have some dietary distinctions there. But we also know that Neanderthals ate plants. They scraped the plaque off the teeth of some Neanderthals. Okay, everyone, when they go to the dentist, they scrape the plaque off your teeth. It's always fun. And in that teeth, the plaque, or what we call calculus, they can extract starch grains. And starch grains are going to be left over from the plants that you consumed. And so there's evidence of some really interesting things like relatives of barley, date palms, legumes, and there's some evidence to suggest that these plants were cooked because the condition of the starch grains is consistent with having been from cooked food. So this has been some pretty interesting evidence of Neanderthal diets, eating both, a lot of meat, but also consuming different kinds of plant species. And this is all very new and novel research. Okay, so it looks like omnivory is the key to evolutionary success. Animals that are omnivorous tend to be the most flexible in terms of their diet. If environment changes, you might be able to shift over to a different food resource. If there's different seasonal differences in what's available, you can eat something different until the next season. You can balance your diet based on nutritional needs, food quality, and based on other dietary choices that are in the environment. So there's a lot of different pathways as an omnivore to obtaining adequate nutrition. Okay, this is where we're going to shift the focus a little bit. So I don't study early hominins. We teach about them. I don't study Neanderthals, but something that we teach about. But I do study native California populations. And so we kind of know that certain things are really important to the diet, especially talking about growth and development, having essential nutrients, having adequate amounts of protein and things like that. And we know that if they were important in the evolution of the human species, they must be important today. And so when we look at archaeological humans from different locations, we often are interested as bioarchaeologists, physical anthropologists that study human skeletal remains, to say something about past behavior. We're interested in different kinds of transitions that we see in prehistory. And primarily we're looking at the last 10 to 20,000 years of time, when we're pretty much just dealing with our species, not all these earlier groups of humans that used to exist. So one of the big transitions that people have looked at is the shift from hunter-gatherer to agricultural types of lifeways. When we use the term hunter-gatherer and agriculturalist as anthropologists, we know that those terms are not very useful because there's an enormous amount of variation in hunter-gatherers, and there's an enormous amount of variation in agricultural populations. But we use it as a general sort of organizational tool to look at some differences in these populations. And so with the end of the ice ages, so the end of the Pleistocene and the beginning of the Holocene, which started 10,000 years ago, we have some significant changes. We have the first evidence of agriculture, in the Near East and other places. And there's different transitions to agriculture in different parts of the world at different places in time. It's a lot more later in time in North America, earlier in time in parts of Europe and Asia. But often the individuals that underwent these transitions, even though they occurred slowly, probably over a number of generations, populations began to become more sedentary. They began to live in one place for longer periods of time. They started to have a more significant impact on their environment. They started to farm, and they started to live in these larger types of settlements. So you have the conditions in place for things like demographic stress. So people basically reproducing at a much higher rate than before. And you have conditions for the intensification of domesticates. On the graph here is the first time stable isotope analysis was ever used to reconstruct diet in archaeology, and it dates to the late 1970s, 1978. And it basically tracked just the introduction of maize agriculture into eastern North America. And you can actually see on the left-hand side, so it starts back on the left-hand side there, you can see that it's basically a C3 diet, and all the way when you get to around 1000 AD, all the way to the right there, you see this big increase, and that's basically tracking a shift from a C3 plant to a C4 plant. And so that's something you could identify in bones and teeth of individuals as they started to become more and more reliant on things like maize for their subsistence. Okay, and that's a big change to go from a more of a hunter-gatherer type of lifeway to one that involved domesticating crops, living in more sedentary villages. Yeah, so it's a bit simplistic of a interpretation that I'm giving, but just to sort of give you the broad strokes. We also see increases in certain kinds of non-specific indicators of health. And when I say non-specific, I mean they're telling us something bad happened, but not exactly what caused it. So like dental caries, we might see an increase in dental disease. Okay, we might see an increase in dental defects that happen due to stress events that happen in childhood, which I'll be talking about in a moment. And we might see some scars of probable anemia on the skull. And we might also see things called periosteal bone reactions, which many bioarchaeologists would like to say are due to infections. And that could tell you something about, how what kind of environment people are living in, and how they're spreading disease amongst the population. Okay, but in California, where I do most of my research, there was no transition to agriculture. In fact, early ethnographers of California thought you weren't going to find anything interesting if you looked at California prehistory because nothing changed. We know that that's not true. We know that there's very significant and dramatic changes in different parts of California all across the Holocene, the last 10,000 years. We know a lot about the late Holocene because that's when we have most of the archaeological sites that have been studied. So the rest of my talk will kind of focus on the last roughly 3,000 to 4,000 years of California prehistory. Told you I was going to talk about 7 million years. I've already gotten through 6 million and some change. So you know you're getting close to the end here. Okay. Now California has been treated as sort of a very unique place, and it is. We all like to live in California, right? And it was treated as sort of a paradise, almost like, California Indians didn't need to farm because they just, if they wanted food, they just grabbed it. And there was, it was basically things were just too good. Why bother? Okay. That was a long-standing sort of idea. But we do know that Native Americans did have significant impacts on their environment. They used fire to burn corridors between village sites. They used fire to manage their landscape like we do such a great job of today, right? There's fires everywhere. Clearing out the dead wood. It creates environments for certain kinds of plants, especially in the spring, to grow. So they basically had depending on what you're talking about, a schedule on how to manage the landscape so that certain resources were available at certain times of the year. And they had a very sophisticated basketry technology, watertight baskets for carrying water, baskets for boiling food with hot rocks. They had very sophisticated types of societies, but they didn't farm. Okay, so in approaching this research, I was interested in whether or not the types of changes that we see in the archaeological record in California, would they look anything like the types of changes we see with agricultural populations? So that's where this sort of investigation began. So everyone's familiar with Central California because we're in it, okay? But that just sort of highlights the archaeological sites where I've studied human skeletons. So I'm going to talk a little bit about their diet based on isotope analysis and a little bit about their health status based on their skeletons. So I looked at populations from the sort of mid-central valley. You can see sites that are near Sacramento and down to around Stockton, and then there's the sites on the eastern bay shore of San Francisco Bay, which I'll be talking about as well. So I wanted to sort of compare and contrast these two different groups of societies through about 4,000 years of prehistory to see if you see similar types of trends to agricultural transitions. Okay, we can't always say that populations that lived there at contact when the Spanish came in the 1700s were the same populations there 4,000 years ago, but the ethnographic territories that where Native Americans lived when Europeans came would fall within the interior or plains Miwok of the Central Valley and the Ohlone Indian tribe of the San Francisco Bay Area. Okay, and this is just showing you again locations of archaeological sites that I'm going to talk about. Now, one of the largest sites, probably one of the largest shell mounds in Western North America, and in the Bay Area, common sites were these large accumulations of shell found around the bay shore of San Francisco Bay. And Nels Nelson did his master's thesis about 100 years ago, where he went and did an archaeological survey of the bay, and he counted about 425 different shell mounds. Okay, so the San Francisco Bay was a densely packed landscape. It was, people have tried to estimate how many Native Americans were in North America, always say that California had more people packed in the landscape than any other place, including agricultural types of places. So lots of people, and you can see from the shell mound, this is probably associated with the village site. There's debate about whether people lived on top of the mound or not, but certainly the mound was used for piling up shell. It was used to bury the dead. There were hundreds, if not thousands, of people buried in the mound, and this was just probably a major cultural center in the San Francisco Bay Area. It's so much larger than any other mound, and it's probably larger than the IKEA that now sits in the footprint of this. If anyone's ever been to the IKEA in Emeryville, this mound is where this mound used to sit, where the IKEA is. And it had a pretty long period of occupation. We have dates from about 2,600 years ago to about 700 years ago. And it's known as the Emeryville Shell Mound. And you can see it's actually being demolished here. It's 1924. It's being demolished by steam shovels. And Berkeley archaeologists tried to salvage human remains and artifacts for the museum. And they did the best they could, but it was awful circumstances, and way before we had, archaeological site protection laws and things like that. But this is just sort of an extreme example of how large these mounds were, and there's probably well over 1,000 individuals that were buried in that mound. Okay, another one, and just to show you that they're not all shell mounds, and a colleague of mine always gets upset when people call earthen mounds shell mounds just because they're in the Bay Area. This one is in the Coyote Hills of Fremont. And there is some shell in the mound, but it's primarily an earthen mound, and it's called the Ryan Mound. And just another example of, there's a lot of variation in the mounds throughout the Bay Area. And then if you go to the interior, to the Central Valley, you have earthen mounds there as well. In many cases were mortuary mounds where the dead were buried. In some other cases, the mounds were also associated with village occupations. Usually on high places, large knolls, above the floodplain, things that you kind of expect. Okay, a number of zooarchaeologists, so people who study animal bones from archaeological sites, have looked at changes in the representation of different kinds of animals through archaeological time. And in the San Francisco Bay, research by Jack Broughton and others have found evidence that the larger, sort of more high-ranked prey, so if you were a hunter, you'd probably prefer a deer over a bunny. They seem to be more abundant early in time, but later in time you see an increased use of smaller types of game. So this is indicated to be the result of resource intensification. So populations are causing some overexploitation of some of the animal populations. And either the animal populations are going down or they're moving away from the people, and people are eating things that are more costly to acquire to get the same kinds of nutrients. And so we see this with just any region where there's significant population growth, there's going to be impacts on the local fauna. Okay, but this could be some kind of indication of resource stress in terms of looking at human skeletal remains from these different time periods. We also know that Native Americans used just an enormous number and variety of plant resources. This is just some of them. I don't know how many of you have eaten any of these. Has anyone in this room eaten something made from acorn before? Okay, there's a number of you. They use all kinds of plants, some for medicinal purposes, some for food, and some for, making baskets and boats and fish nets and things like that. But all these plants that they consumed were processed for specific purposes, but they're all C3 plants, and so we don't have anything like a maize that sort of pops out sort of chemically or isotopically that looks really obvious, okay? But what we do have is marine foods, and I'll get to that in a minute. We know that sometime during the late Holocene, acorn storage economies became probably intensified, and so in California, one of the staple crops for many tribes was acorns. They collected them in the fall. They stored them for 1 or 2 years in these large storage structures, and they had access to those throughout the year. Acorns are a good nutritious source in terms of their carbohydrates and their fats, but not necessarily very good in terms of their proteins. And they're a real bear to process, and so here's a Native American woman here using a leaching basin. I don't have time to go through the whole process of how you make acorns palatable. You have to pound them. You have to crack the nut open. You have to cut the skin off. You have to pound the nut meat. You have to heat up rocks in a fire. You have to boil water, and then you have to leach out the tannic acids. If you want to know the whole story, I have a book on the entire process. It's very, very labor intensive. And so some archaeologists have read this as these acorn intensive economies are becoming, more widespread during the late Holocene because there's more people to feed. And this is a good sort of buffer resource food. It's not the healthiest if you eat that as your main staple food, but it certainly can support a much larger population. And as well as all those other different plant resources that people were consuming. Okay, so now we're to the isotope part here. And so this is just a food web reconstruction. So for stable isotopes, we usually look at carbon isotopes on the x-axis there, on the bottom, and then nitrogen isotopes on the y-axis there. And basically, the more you are to the left of the graph, the more that your diet is coming from C3. And the more that you're to the right of the graph, the more that your diet's coming from marine resources. So if you just sort of look, C3 plants and terrestrial herbivores like deer and elk are on the left side of the graph, and then things like marine mammals, marine fish, and shellfish are to the right of the graph. And then nitrogen follows this, what we call a trophic level effect, that the herbivore is enriched over its diet by a value of about 3. The carnivore that eats the herbivore by another 3, and so on. So you can see those marine mammals are way at the top, and marine food chains are longer, and there are more, and there's more trophic levels, so that's why marine mammals have such high nitrogen values. The only thing that's higher than a marine mammal are humans eating marine mammals. So we don't see that too often as a primary dietary resource. So this is some data from my dissertation, and this just shows you some dietary variation in San Francisco Bay. And I'm going to use my other clicker here so I can have a pointer, if I can get this one turned on. Yep, okay. It's going to work in 3 seconds. Yes. Okay, if you can see that. You can see that these are people in the San Francisco Bay, and you can see that some of them are way up here, indicating that their diet is coming primarily from marine. They're eating lots of marine protein, so like fish and sea mammals and things like that. And you see as you go down here, more and more terrestrial foods being consumed. Part of this is geographic, so some people live in the more North Bay near Berkeley, Emeryville area, and some live more in the South Bay near Fremont, and so there's some differences in the environments that can account for some of this variation. But we also see that even people at the same site are sometimes eating different kinds of foods. And if we bring in the Central Valley populations here, we can see that they're eating from a different food web than the San Francisco Bay people. They don't overlap at all. And it was really nice to see this because we have, basically these two different populations of people eating completely different kinds of foods. Okay. All right, so just to sort of summarize, we can see that early in time, these early Bay people were very marine dependent. We're eating lots of marine foods, going out on the bay of these little Thule balsa rafts, which I don't know how they navigated on the bay in these things, but they did. Okay. Later in time, a more mixed diet where they're clearly eating more C3 plants and also meat from animals that eat C3 plants, and less marine, but they're still utilizing the bay, just not as much. And then in the valley, we just don't see a lot of variation over 4,000 years of time. Everyone seems like they're eating the same kinds of foods over time. But we do see that their diet seems to be, their protein seems to be coming primarily from freshwater fish. And that's something that's kind of interesting because people had assumed that salmon were very important in the diet of people from the Central Valley. And they probably were the farther north that you go. And we'll be testing that soon. But here we're not really finding any evidence for salmon consumption. And that's really interesting because the ethnographic record talks about how important salmon were to people in these exact locations. And so it might be that the salmon were eaten in more troubled times after Europeans came and disrupted the cultures of Native Americans, but were not as important early in time in that location in prehistory. And if they were eating a lot of salmon, they should be actually up here. And you can see they're not even close. So this is definitely something where isotopes can shed some light on the ethnohistoric record. Okay, we do see there's a lot of dietary diversity in the bay. I've added a lot more sites with working with various graduate students over the years. And I want to hone in on these people that are way down here in this little box there. What do you think's going on with those individuals? Their nitrogen values are very low. Anybody want to take a guess? Nobody? Okay. They're vegans. Okay. There's basically almost no trace of any kind of animal protein. They're basically at the same trophic level, just barely above plants. Okay. And I found this really interesting because they're not that far from the bay shore. And, it's a small sample, but this is really interesting that, looking at all these different societies, you found a group in the Santa Clara Valley that don't appear to be eating any meat, or they're eating in such a small amount that it doesn't register isotopically. I think that's really interesting. So this is just opening up a whole nother can of worms to sort of look at. Okay, so that's the diet component. I wanted to sort of close talking a little bit about some expectations for health. We've kind of established what people were eating in these different regions. Let's look at how their health changed. And because bone can only respond in a limited number of ways to disease, we often look at these non-specific indicators of health that I mentioned earlier. So one of them is developmental stress defects that you see in teeth. One of the more popular or common ones looked at are called linear enamel hypoplasias. Just a fancy way of saying it's a growth line that you can identify in a tooth. And these are happening when the teeth form. And so when you're a child and your teeth are still forming in the gum line, some stress events happens. It could be malnutrition, it could be disease, but your tooth enamel doesn't completely form, and you end up with basically a band that's thinner than the surrounding enamel. And we can estimate what age that happened to you and things like that. But it's used as an indicator of possible childhood or stresses. Also, we can look at possible evidence of anemic response. We have these lesions that happen in the cranial vault called parodic hyperostosis. Sorry, I'm using all these big words. Just basically, you see a lot of porosity on the skull or in the eye orbits in the case of criborbitalia. And traditionally, these are viewed as a response to some kind of anemia that individual experienced, probably in infancy or early childhood, when there's still a lot of red blood cell production occurring in the skull. Okay, and then the third one is just looking at possible evidence of bone infection and looking at evidence of tibial periosteal reactions, which are just lesions on the lower legs that could be telling you something about infection, although there are other things that may cause those as well. So in prehistoric California populations, I examined these defects in both the samples from the San Francisco Bay and in the Sacramento Valley. I found totally different patterns in the two regions. There is a significant increase in the Sacramento Valley. The hypoplasia defects affected about 14% of individuals early in time, and it jumps up to about 42% and 50% in the middle and late period. So we do see the sort of increase in these stress lesions on teeth. In the San Francisco Bay, of course, you can never have things work out perfectly. You see a decline that's quite significant during the same time period as you see the increase in the Sacramento Valley populations. Okay, and I'll address that in a minute here. And then so lesions increase in the valley, suggesting more stress, and they decline in the bay, suggesting less stress. Okay, if we look at cribororbitalia, so the lesions in the eye orbit, and also product hyperostosis, the lesions on the cranial vault, as possible indicators of anemia. The current interpretation is that to really have an expansion within the cranial vault or an enlargement of the marrow space there due to megaloblastic anemia, those tend to be associated, if it's not a genetic thing, like a sickle cell disease or something, it's associated with deficiencies in things like vitamin B12 and vitamin B9, so cobalamin and folic acid. This could be due to inadequate diet, so people aren't getting these proper nutrients in their diet when they're an infant or a child, or it could be because you're riddled with parasites, and the parasites are preventing the absorption of those into to your body, or having things like extensive bouts of diarrheal disease where it's being lost that way, which is very common in developing nations that are experiencing things like poor sanitation. So let's look at the pattern here. This is just pointing again that sieve-like pattern in the eye orbits. We see a significant increase in the Sacramento Valley between the early middle period. The lesions are very low, and they really seem to jump in the late period. In the Bay Area, there's no significant changes. It looks like there's an increase, but it's not significant, so statistically. So again, some divergent patterns between the valley and the bay. That's just showing you, you probably can't see it that well in the photo, but product hyperostosis, which is the porous lesions here on the skull. Just so you can see, it looks a lot worse when it's very active. That's a healed form. You can see in that x-ray that's an individual who has a form of anemia, and you can see it has that hair on end, and that's all of that sort of bone that's being laid down, and the marrow space in the skull is enlarging due to extra production of red blood cells. It's called marrow hyperplasia. Doesn't look like something you'd want. Okay. But let's look at the pattern here, and we see that there's no increases through time in the vault lesions in either location. But one thing that stands out is the San Francisco Bay is a much higher prevalence over here than what you see here in the Central Valley populations. And that seems to be a theme that almost anything you compare, the Bay Area is more prevalent. They have more of these stress indicators on their bones and teeth, but they don't change through time. They're just high all the time. And in the valley, most of the indicators show an increase. Okay, and then these possible evidence of leg infections that we call periosteal bone reactions. Lots of things can cause these, but often if they affect both legs, they're more likely to be due to infection. And these can happen, they seem to happen more commonly on the lower legs, and that's a more common site of injury if you've ever bumped your shin on a coffee table or something like that. But they seem to be more susceptible to getting these infections, and so they tend to be studied more in those bones than in other bones. But these are commonly associated with things like higher population density, so you have more people in the same location. You have the conditions for poor sanitation, greater person-to-person contact, and so with increasing populations in both of these regions during the late Holocene in California, you might expect to find a higher prevalence of these lesions. Okay, and in the Central Valley we do. I told you it was a theme. There's only one individual that had a lesion in the early period samples, and then you see the significant jump in the middle and late period. So it seems like it's an increase in the prevalence of these infections, at least in the samples that I examined. And then in the in the bay, you see no change. It looks like, again, like there's a change from maybe the middle period to the late period, but it's not significant. So we're not really seeing any statistical evidence to indicate it changed. But again, it's generally higher, especially during the late period in the bay, than in the Central Valley. Okay, so we are coming to the end, and bear with me. So just to sort of summarize, if we look at the San Francisco Bay, the prevalence of these conditions is relatively high. So it does suggest that infants and children likely suffered quite a bit of dietary and nutritional stress and disease, and it was higher in general than in the populations from the Sacramento Valley. So we do see some sort of regional patterns there. We only see a significant decline in the maxillary canine hypoplasia defects in the teeth in the Bay Area. So this doesn't really give us much temporal or time information about how disease changed in that region, but it does indicate that it was, there were significant health problems in children. And of course, we're looking at these lesions in adults that survive they survived their childhood, but they still bear the scars of these lesions. In the Sacramento Valley, it does seem to support sort of my central hypothesis that you'd expect to find more indications of poorer health and nutrition associated with increasing population, increasing sedentism, more people on the landscape, and more permanent settlement in the Central Valley. And this seems to be mostly supported by the lines of evidence that I examined. And again, just reiterate that the Bay Area seemed to have significantly higher lesions than the Central Valley populations. Okay, so in terms of looking at this as a transition, like the transition to agriculture, it does seem to support some of the basic notions. However, I would like to point out that when we look at transitions, not all of them were as bad as others. And so in many areas of the world, you don't see declines in health associated with those kinds of transitions. In Central California, it looks like these transitions varied between regions quite a bit. So there's a lot to think about here, but we're trying to explore these big questions in anthropology and bioarchaeology, but we're using very imperfect and limited lines of evidence. But my final thoughts are that clearly omnivory is a hallmark of the human species, and it's been with us for a very, very long period of time, and having this enormous dietary diversity and flexibility. Looking at specifically the research I've been doing in California, the diet and health patterns in Central California seem to conform to many agricultural transitions in other regions. Also, just as a sort of a summary statement, examining broad patterns of ancient human diet and nutritional status provide a window into the biological consequences of subsistence change in different parts of the world. So this is clearly, going to continue to be a major area of interest by bioarchaeologists working in California and elsewhere. I'm going to end on just want to thank my 2013 Paleonutrition Graduate Seminar. It's been a lot of fun, and I know a lot of you were there. And Heather, I'm sorry, you were out of the room when we took the picture, and I couldn't Photoshop you in. I just want to, the class has been great, and we've covered a lot of the topics that I talked about today, and I've just learned so much from the class, and it's just been a lot of fun. And I just want to acknowledge College of BSS, Dr. Eddie Vela and Dr. Teddy DiLorenzo for inviting me to speak, and also for their support. Kathy Castleberry, who's done so much to help put this on and coordinate everything and remind me where I'm supposed to be, even though I always go to the wrong place. And I want to thank my Department of Anthropology, and Dr. Nettie Martinez for all their support. I want to thank Ron Lindsay and Emily Sinibo from Graphic Design and Web Services. They put the wonderful flyer together on the right-hand side. I also want to thank Alan Leventhal, archaeologist at San Jose State University, and also the Muwekma Ohlone Indian tribe that he works with, who have been supporters of this research, and current and past students and fellow collaborators. Thank you.
Hi. A couple of questions. One is, why wouldn't salmon be in the diet of the Central Valley?
Why wouldn't it be?
Yeah. I mean, what hypotheses do you have that they weren't eating salmon?
I don't know if I have a hypothesis. There's not a lot of faunal evidence for it too, and people would say, well, the bones just don't preserve very well, or different kinds of things like that. That part of the valley is not an optimal place to catch salmon because the delta is so massive, and it looks like people were targeting more freshwater fish. But if you go up to like Redding or someplace like that, they built weirs or fish dams across the entire river where they could actually monitor, salmon and stuff like that. So it could just be there's places that are geographically preferable to mass capture salmon, and it just looks like that region really wasn't. But perhaps when after Europeans came, there was significant disruption to native societies, that they were eating more salmon at that time than they were in the past in that region. So we're going to test this looking at some sites in Shasta, or a site in Shasta County, and some of my collaborators like Jim and Denise are going to be working on that, as well as Frank. And so hopefully we'll identify a salmon signature there, because there's populations in British Columbia. They're located really far inland, and you can see they look like they're marine foraging, but they're inland, and they're located on salmon streams. And so you kind of know what the salmon are going to look like when you see it. But yeah, I don't have a great overarching explanation. It's a lot of fish biologists have been noticing that this, maybe what they thought all along isn't correct. So.
Hi. Over here on your right.
Hello.
Thanks for the great talk. Nice to see you. I'm trying to wrap my head around something that you mentioned earlier in your talk, Eric, and that was the trend in thicker tooth enamel as a very general pattern in human hominin evolution. But then shortly after that, you mentioned that modern foragers consume nutritionally dense diet, 30% animal protein. So it seems like maybe they're going in different directions. Are these foragers, are they an anomaly, or is this part of
no,
a
this is
pattern?
and there is quite a bit of variation. There's some populations that they're getting about 50% of their macronutrients from protein, but they're living up in Alaska or something like that. Oh. But and obviously modern hunter-gatherer populations are very different than they were even 50 years ago or 100 years ago. But some of the estimations that they're getting about that much protein, that might include insects as well, which I know it's up your alley.
Sometimes.
Good source of protein. But it seems to be higher than you would maybe expect. But the figures are kind of aggregated from a number of studies, so there's some range there.
Thanks.
Thanks for the talk. Seems, if I understood correctly, the suggestion in your data that the Bay Area remained at fairly high levels of nutritional stress through all three periods. The Central Valley moved in much of the data from lower stress levels to higher stress levels. Maybe could you comment further on what led to those high stress levels potentially in the Bay Area and the processes that might have brought the higher stress levels into the Central Valley?
This is certainly something I've been thinking about quite a bit. It could have to do with land mass, and so the Central Valley is enormous, and the population was much higher. It was even the Sacramento Valley population was estimated like 52,000, and the Bay Area one about 10,000. But just in terms of maybe persons per square mile. And so the Bay Area might have been more densely settled in terms of it's a smaller space. So that's one idea. And maybe if we were to look at skeletal samples earlier in time than we have available, we would see lower levels of stress. And so it could be that, the Bay Area was a great place to live. People packed that landscape first, and then the valley took a little bit longer to sort of reach more of a carrying capacity or something like that. That's just, I'm just talking, but there's got to be an explanation for it. But that's my thoughts. But
what would you expect from a diet of grasshoppers in the Great Basin compared to pronghorn? Would that signature? Would you expect that signature to look the same?
It's hard to answer that. Grasshoppers are fairly, it's a good source of protein, and it depended if the grasshoppers were eating C3 plants or C4 plants. If they're eating C4, then if you were eating the grasshoppers, you're also eating the C4. So that would differ quite a bit. I think that they would be different, and I think there's some data that Margaret Schoeninger and some others did in a volume on that in part of the Great Basin, but I'd have to look at that. But I'm not sure off the top of my head. But grasshoppers were consumed here in California by many groups as well, so I don't know what their isotopic signature is, though.
Hello. Are you here?
Oh.
I'm curious if you could go over how the trends are continuing on through similar cultural demographics over the last 100 or 200 years, the ones that you talked about. I assume that you're talking about the last 4,000 years kind of through that advent. So.
Yeah, I mean, one of the things I left out was what happens at 1769 in the Bay Area, and it's Spanish come, and they come into the Bay Area in 1769. They missed Monterey Bay. They were lost, and they found San Francisco Bay, and all the people already living there. But there was a mission period that ensued after that, and so groups of people, native groups, were assimilated into different missions and dying in high numbers from infectious diseases like syphilis and other kinds of diseases that were brought in by Europeans. And so their lifeways were completely devastated. I did do a study from Mission Santa Clara, and you actually see some evidence of Native American neophytes eating corn. And I think that's so funny that the Spanish basically acquired corn from Native Americans in other parts of the U.S. and introduced it to California during the mission period. And you can actually see the corn signature, the C4 signature, showing up in some of the mission Indian groups. And some of them seem like they're eating more of their traditional native diet by comparing them with prehistoric groups from the same area. So it's kind of an interesting window into that kind of culture context situation. Did that answer your question?
Yeah, and maybe more so in areas that weren't affected by an introduction of human disease, and more so areas that you could have more of a touchstone and a continuous culture to reference through that.
I know that there's still several Native American groups that are they have completely maintained aspects of traditional lifeways, like in the coast, and salmon's still an important part of their diet. They smoke salmon and things like that. So I think depending on what part of California you go to, you'll find a very vibrant, engaged Native American community that still try to maintain aspects of their diet and language and things like that.
Thank you.
Okay, one more question. You get a microphone.
Thanks. Yeah, I just wanted to ask, you talked about the thickening of the tooth enamel in response to dietary pressures, I guess. I think you said something about rougher food. Could you
More coarse foods?
Yeah, coarser food, like what?
There's been all this research on, you can look at how teeth wear, and you can look at the plane of the wear and things like that, and then you can look at it under a microscope. It's called microwear, under a scanning electron microscope. And in our seminar, we read a lot of papers about that, and it really seems like we don't really have a very good notion of the specific foods that they eat. The isotopes seem to tell us a little bit of something, but there's a lot of things. Having hard tooth enamel is good for certain kinds of coarse food. So certain kinds of tubers, for example, that's a big one that gets brought up. Certain kinds of nuts and things like that. Having harder, tooth enamel, where chimpanzees have thinner tooth enamel, and when you look at humans through time, the tooth enamel seems to get thicker as time goes on.
Hi, Eric. I enjoyed your presentation. And I was just wondering, if you had the good fortune of getting a little sample of everybody's bone in this room and ran your analyses.
It would do it from your hair.
Oh, you can do it from the hair. Okay, then you don't need the bone. But
Less intrusive.
Far less
Easier to get through the IRB.
Okay, but how would the graph look in terms of diet? Would you just? I don't know how it.
Most
would
of
look.
us would be shifted towards C4 because of the amount of corn products we consume,
Oh.
and that's been tested because different people have been testing isotopes in hair, trying to see what people are eating culturally, and you can still distinguish between different groups and different parts of the U.S. and different cultures from with the isotopes in their hair, but you'd be surprised at how much C4 is actually represented in your carbon isotopes in your hair, because high fructose corn syrup or corn just seems to get inserted into so many different processed foods. It's kind of mind-boggling. And when I was a graduate student, we tested our own hair in a mass spectrometer, and there was a student who was from China who ate nothing but a C3 rice diet pretty much most of his life. He really liked McDonald's and a lot of C4 in McDonald's foods, and he could actually see a shift from how long he had been here to the more C4 he'd been eating since he was in the U.S., and I thought that was kind of telling. All right, thank you so much.